Antifungal resistance is no longer a rare clinical problem. It's a growing global threat. Let's break down exactly how fungi outsmart our treatments. There are 4 main resistance mechanisms. First, target alteration. Many azoles, like fluconazole, inhibit the fungal enzyme lanosterol 14, A-dimethylase, which is essential for ergosterol synthesis in the cell membrane. Resistant strains, such as Candida albicans, can mutate the ERG11 gene encoding that enzyme, reducing drug binding without compromising its function. Second, efflux pump overexpression. Certain fungi overproduce ATP-binding cassette transporters or major facilitator superfamily pumps. These act like molecular bouncers, actively ejecting the drug before it reaches inhibitory concentrations inside the cell. Third, biofilm formation. Biofilms, especially in Candida species, create a protective extracellular matrix that limits drug penetration and allows dormant persister cells to survive therapy, leading to chronic or recurrent infections. Fourth, bypass or compensatory pathways. In echinocandin resistance, for example, mutations in the FKS genes alter beta-1,3-glucan synthase, the drug target. Some fungi also upregulate alternative cell wall synthesis pathways to maintain structural integrity despite the drug block. Clinically, this means infections by resistant fungi like Candida auris or Aspergillus fumigatus can fail standard treatment, requiring combination therapy or newer agents such as ibrexafungerp or olorofim. The key takeaway: fungal resistance is driven by genetic adaptation and selective drug pressure. Judicious antifungal use, accurate susceptibility testing, and ongoing drug development are our best defense. If you found this breakdown useful, join us next week for another deep dive into cutting-edge microbiology.